disappearing now. Too far back. Start in 1 minute.
You're making me laugh.
I once did that. I had a talk. I was giving this like seminar series, and we had some point where we had to taste wines, and and it was a forced like preference tasting, and like you had a number of wines. You had to tell me which was the worst and what was the best. It wasn't super scientific, but it was scientific enough so we could tell which wines people really didn't like. Anyways, during the tasting and answering period, I played the Jeopardy music as a background. It was pretty funny.
Merv Griffin.
That's it, copyrighted. All right, I'm going to begin. Hello, everyone. My name is Darren Michaels. I am the host of this Scott Labs series where we discuss interesting topics, try and answer questions related to wine, cider, or specialty beverage production. From yeast and nutrients to stabilization and filtration, we dip into the Scott Labs vault of over 80 years of experience to bring you the latest in research and technical applications. My name is Darren Michaels. I'm a Scott Labs fermentation outside technical expert that works out of the Pacific Northwest in the U.S. And when I thank you all for attending and thank you all for taking time out of your day to join us. Today is part 2 of Dr. Hall's talk about working with our budding geniuses, yeast management and selection to maximize wine quality and drive wine style. If you have missed part 1, please go back and listen to it. It is on our website. Just Google Scott Labs webinars. It should show up. Today is when we take the principles from part 1 and put them into practice. I also again want to recommend that if you don't have our current 2020 winemaking handbook, that you download a version from our website or call the office. They might mail you one. Just Google Scott Labs 2020 handbook and you should be able to find the link. Before we begin,
Oh, Darren, you are frozen.
some quick webinar tips. Make sure you can see the audio setting. Am I frozen? Can you guys hear me now? How is it now? Can you hear me now? Am I back? Yeah. If you have, before we begin, some quick webinar tips, make sure you can see the audio settings on the lower left and along the bottom, the raise hand Q&A buttons. As usual, these will be the main channels for communication. If you have any questions that occur to you during the talk, feel free to post the question on the Q&A section. We will try and answer it after the presentation. Your questions will be anonymous. I'll also be looking for raised hands if you'd like to ask your question live. Just so you know, we will be recording this webinar. We will be making it available soon after today. And then you can find those at Scott Labs. You could Google that as well, Scott Labs webinars. I've said this before. Sometimes it can feel that with winemaking, we're herding cats. Last week's talk was about understanding kittens, and today is about learning how to feed them. So without the further ado, I'd like to hand over the microphone to Dr. Nichola Hall. Nichola, how are you?
Hey, thanks, Darren. Good morning. And Darren and I are just giggling offline here, so just ignore us.
Giggle away.
So as Darren mentioned, we're going to just kick off from where we started last week with our budding geniuses. So last week we spoke about what yeast are, what yeast were, how yeast grow, what they need to grow and survive, and how that information applies to winemaking, and then selecting your enological yeast for success. And we used our Scott Labs handbook as a driver for that. So we kind of walked through the different sections. So this week, we're just picking right back up. We're going to talk about yeast dosage, yeast handling, and using yeast to drive wine style. So there's no point in spending all the time looking at selection, making sure the yeast that you choose is suitable with the juice or must chemistry with your winemaking style, and you just don't use them to their maximum benefit. So that's kind of what this week is about. So let's kind of jump right in without further ado. Talk about our yeast dosage. So why do we use 25 grams per hectoliter? Now, if I had $1 for every time a winemaker told me, you just tell me that, Nichola, because you'll sell more yeast. I would maybe wouldn't have to do webinars, wouldn't have to work for a living, but it's not true. If you actually underdose your yeast, we sell you more because you've got a good chance of sticking your fermentation. So why do you use 25 grams per hectoliter of yeast? Well, you lower that yeast potential for osmotic shock or sugar shock, okay? You're going from a nice, active dried yeast, you're going to a high sugar environment, and you've got something in between. So enough yeast there protects against that initial shock. When we spoke about the growth phases last week, we spoke about the lag phase, and the duration of that lag phase was dependent on the growth conditions, the number of cells that were there. So 25 grams per hectoliter will shorten that initial lag phase, which means that you will get up to 4 to 5% alcohol much quicker. So when we said last week that there was about 80 different genre yeast, 20 of those different genres are associated with the winemaking environment. A lot of them do not have anything of interest from an enological perspective. A lot of yeast at the beginning of fermentation, native yeast I should say, can produce ethyl acetate and acetic acid. So if we shorten our lag phase, we get up to 4 to 5% alcohol as quickly as we can. So we're getting into that growth phase. We're really starting to suppress those native microflora and letting our Saccharomyces do what it's supposed to do, which is take over the fermentation. And we have that faster onset of fermentation. So the shorter the lag phase, the faster onset and fermentation, the faster the yeast get through that yeast growth curve, then the shorter the fermentation length overall. And as I mentioned, the shorter that lag phase and the faster the onset of fermentation, the lower the VA. Interestingly, when we look at 15 grams per hectoliter versus 25 grams per hectoliter in different conditions, we see that at 25 grams per hectoliter, we end up with a lower final RS under different conditions. So we're actually using enough yeast to help secure our fermentation. And our yeast is just much healthier overall. So we're going to make sure we have the correct amount of yeast there to begin with. So we've selected our yeast. We know how much yeast to add. How should we handle our yeast? So how do we handle our Saccharomyces? So we're going to go through what is an active dried wine yeast? What is the rehydration protocol? What are the consequences of mishandling and the importance of homogenization or mixing? Okay. So when you get your active dried wine yeast, you're getting a little 500 gram pack, 10 kilo box. If you're on the Lallemand side, a 250 gram pack, or a kilo if you're on the Anchor side. But really those yeast, we have removed the water during the dehydration process. That is a desiccation process. And that water deficit leads to an arrest of the cellular functions. So there are a lot of cells in that 500 gram pack, about 10 to the 10. And our job during the rehydration is to replace that cellular water. So we're going from a dehydrated to a rehydrated phase. Now, the rehydration itself, when we add back that cellular water so that we have a reinstitution of cellular functions, it is a biphasic process. So it's a two-phase bifurcated process. The first stage is the rehydration of the plasma membrane. And that takes place during the first 20, give or minus a few seconds, 20 seconds. And that plasma membrane, remember, is the site of transport for our sugar, our nutrients into the cell, our alcohol, our other interesting molecules out of the cell. So we need that membrane to be optimized. So that semi-permeable, highly specific plasma membrane is going to be rehydrated during the first 15 to 20 seconds. After that time, then it is going to be more discerning what can come into the cell. And that's when we really start to have the rehydration of the intracellular components. So all of those organelles that we discussed in week 1. A proper rehydration has a very positive effect on the viability and the physiological state of the cell. So the viability, how many living cells there are, and the vitality, how many cells are metabolically active. It has, because it impacts the viability and vitality, it's going to modify the fermentation behavior. So particularly the lag phase. So a good rehydration minimizes that lag phase. When we look at all of the things that we can do during rehydration, when we look at water quality, water quantity, and with that, we're looking at hardness level. So how many ions are in there? When we look at aeration, agitation, the presence of sugar as a carbohydrate, and temperature, we see that temperature is the most important aspect. Temperature above 109 degrees, 110 degrees, has a negative effect on the cell viability. Once we're at, above that temperature, we're going to get to sublethal temperatures for Saccharomyces, with 115 getting to lethal. When we're below 30 degrees Celsius, which is 86 Fahrenheit, those fatty acids, those phospholipids that give the stability to the plasma membrane will crystallize. So they'll change the ratio between unsaturated and saturated fatty acids, and the membrane will be damaged. And the damaged membrane means we have a loss of internal components, we have a decrease in fermentative capacity, which means our vitality is going to be impacted. So the temperature is the thing that you want to take care of the most. It's so much more important than aeration, agitation, water quality, and sugar content. So what does a proper yeast rehydration protocol look like? And again, I've taken this directly from page 36 of the 2020 handbook. So step 1 is you're adding your rehydration nutrient, your GoFerm or your GoFerm Protect Evolution. And that goes into the water around about 110. Now we want that water to be hot so that the sterols that are involved in our rehydration nutrients go into the proper shape. They retain their conformation. We put that into the water first because I just said rehydration was a biphasic process, and the first 20 seconds the cell cannot discern what it's taking up. So once you put your yeast into the water, which has now dropped down to between 98 degrees and 104 degrees Fahrenheit, when the yeast goes in, it's going to take up all those goodies from our rehydration nutrients, our vitamins, our minerals, our unsaturated fatty acids, our sterols, and they are going to become part of the strengthening of the cells. We wait about 20 minutes. We can actually wait up to 30. We don't want to wait much more than that because our active dried wine yeast has some storage carbohydrates. So we said that yeast are chemi-organotrophic, which means they need to be supplied with their carbon source. So they actually have some storage carbohydrates in them during the active dry form. They have trehalose and glycogen. And after about 35 minutes, okay, it's always a kind of a very wobbly time. It's not like a stringent time. Those storage carbohydrates will be depleted, and the cell will look for more and more sugar. So we want to, once our yeast goes into the water, not leave it in there too long. So 20 minutes is a good rule of thumb. Definitely no more than 40. Start to worry when you get to 30. If we're to take our beautifully rehydrated yeast, which we've replaced the cellular water, we've got the activation of about 3,000 genes straight into our juice or our must. That is way too much of a differential with regards to temperature and sugar. So we want to do a step in between. We call it an attemperation or acclimatization. And that's where we take some of our juice or our must, we add it into our yeast slurry, and we will drop the temperature. It's 10 degrees Celsius, and we hold it for 15 minutes. 10 degrees Celsius is about 18 Fahrenheit. So the rule of thumb is drop the temperature 15 degrees and hold it for 15 minutes. That's an easier way, I think, to remember it. After your 15 minutes, when the yeast has been subjected to some sugar, it's going to start activating its sugar transport proteins. It's going to have been given a little bit of nutrient from that original juice mass, start to activate other transporters. You want to measure the temperature again, and if you're within 15 degrees Fahrenheit of your juice or must temperature, you could go ahead and inoculate it. If you are not, you must keep repeating this. So it's a stepwise acclimatization from, for example, 100 degrees to 85 degrees to 70 degrees. Most things need 1 or 2 acclimatizations, and then you are good to inoculate. And again, there is a video on our website on yeast rehydration, as well as the full protocol, page 36 of the handbook. And that's for Saccharomyces. Remember, your non-Saccharomyces has to be rehydrated at a much lower temperature. For non-Saccharomyces, for things like Flavia, Biodiva, Gaia, and Lactea, they must be rehydrated between 20 and 30 degrees C, which is 68 to 86 Fahrenheit. Now, sometimes we look at foaming and we get really excited, but foaming doesn't mean much in the grand scheme of things. Foaming is not an indicator of viability. Some yeast will foam a lot, other yeast foam very insignificantly. And so, in fact, some yeast like Osmanhausen doesn't even look like it's rehydrating at all. So you want to use some sort of timer and a thermometer during rehydration and not go by look and feel. Okay, so rehydration is a very important process for fermentation success. So what if we don't? What if we decide to ignore some of those steps, or we don't train our staff so they understand the importance of these steps and we mishandle our yeast? Well, we have our cell wall and our plasma membrane structure is going to be compromised. We're going to have intracellular damage, which means our viability and our vitality of our cells are going to be impacted. We are going to have a longer lag phase, and those storage carbohydrates, that trehalose and glycogen, can be depleted. And Michelle, you have a question?
Oh, does somebody have?
So the question is, how vigorously do you want to agitate the GoFerm into the rehydration water? Is it important to get rid of any clumps? So GoFerm and GoFerm Protect Evolution are fully autolyzed inactivated yeasts that contain a high level of vitamins, minerals, sterols, and saturated fatty acids. So you don't want to have any clumps there. So the hotter the water, the easier it is to disperse. It is sticky, but that stickiness is quality. So try to make it go, it won't go fully into solution, but try to get it with no clumps in it. Okay, hope that answers your question. If
Yeah,
not,
the second part, I didn't,
again.
yeah. we were going to wait until the end, but I'll just go ahead and ask you. It said, yeah, how vigorously do you want to agitate the go ferment in the rehydration water? And is it important to get rid of any clumps? How important is that?
Yep, so we've just addressed that. It is
Okay.
important. If it's clumping, your yeast doesn't have access to it, right? So try and get it into some sort of a slurry. We say for your 20 times the yeast weight in water when you're using your rehydration nutrient. So for every gram of yeast you're using, you need 20 milliliters of water. If you've got a little bit more water, it's okay. You don't want to have much less, or you've got too stodgy an issue. So let's move on. So we've got our yeast, our dosage calculated, our rehydration, it's acclimatized. Now we have to actually add it. Now, I'm not quite sure what happened. A few years ago, people started telling me, well, I just add it into the corner. It's Russian River Pinot Noir, Nichola. I go, okay, well, the yeast is not aware it's Russian River Pinot Noir. The yeast doesn't know it is a vineyard designate. The yeast knows it's in an environment with sugar, nutrients, and other factors. And it wants to take up the sugar, nutrients, divide maximally to take over and be the dominant organism, fully implant. Now, in order to fully implant, we want to make sure that once we've added our yeast, we mix it in there thoroughly. Yeast in that cell membrane has a lot of transporters, and they have different transporters for different nutrients. We have things that we call high affinity transporters, and we have low affinity transporters. The high affinity transporters are what are operational when the yeast sees low levels of nutrients. And the low affinity transporters are operational when the yeast sees high levels of nutrients. So it's like a replete or depleted situation, and you'll have different transporters operational, which means the yeast is working differently at the genetic level to change its dynamic flux to accommodate. So if you just inoculate your yeast into one corner and you don't fully mix it in, you're going to have so much yeast in one area, it's going to take up all those nutrients, and then it's going to actually run out of nutrients, and it's going to change its metabolism. Likewise, if you don't fully mix in the yeast, then you have all your Saccharomyces in one area and all the native flora in other areas. So you're not working to outcompete. You're not using your yeast, your Saccharomyces yeast, I should say, as a bioprotector, which is a buzzword in our industry, which essentially means that you have one organism controlling other organisms and minimizing those other organisms' impact. Okay, so it is really important to do. And it's not just in the US that we're talking about mixing. The best paper from the American Journal of Enology and Viticulture, which is the journal from the ASCV, their best paper last year was by Dominique Lerner's group out of Neustadt or Giesenheim, I've just forgotten, the German group. And they looked at the distribution of yeast cells, temperature, fermentation byproducts, and white wines in different shaped vessels, and demonstrated really nicely that when you don't mix your fermentation, your metabolic profile is different. And in these non-mixed fermentation, your acetaldehyde is higher, your pyruvic acid is higher, your acetic acid is higher, and these are all fermentation inhibitors. Okay, so mix your yeast is the short story to a very, very long answer. So we've got our yeast, we've got our dosage, we've got our rehydration and our mixing. Now, the yeast, as we said last week, Cerevisiae and its friends are our enological yeast of choice. There's about 300 different strains available. And we spoke last week about the technical attributes and the sensory contributions, and really how we can utilize this diversity. And really what we're talking about is the yeast metabolism, how we can optimize the yeast metabolism. So yeast, we can talk about is primary metabolism, and that is when they're using the nutrients for growth, cell division, and really survival. And they're producing primary metabolites like ethanol, glycerol, acetaldehyde, and acetic acid. These are our neutral strains. Sugar plus yeast gives us alcohol, CO2, and these other minor components. Not many yeast now in our isolations are really neutral fermenters. We're really looking at strains that excel due to their secondary metabolism. And yeast that are excelling with secondary metabolism is our yeast strains that are producing or revealing compounds that are very interesting from a wine sensory standpoint. So we do sugar to alcohol, or we do sugar to alcohol with all these other really cool things. And remember, your fermentation-derived compounds are really making up the largest percentage of these aroma compounds in wine. This is what makes wine so special, is these secondary metabolites. So kind of, we know that there's a lot of secondary metabolism going on, right? Because we smell it, we taste it, we feel it. Wine is a sensory beverage. We also know through our chemical analysis that when we look at the volatile compounds in juice, and we look again at the volatile compounds in wine, they're changing. And they are changing due to the presence of our friends Saccharomyces cerevisiae, our little bacteria, Inococcus ini, as well as these days our non-Saccharomyces. So although we're smelling things and feeling things and tasting things and looking at chromatographs, we're really trying to put the link now between the two and saying, okay, why am I seeing this different profile? And how can I understand what is going on at the metabolic level that I am seeing said compounds being produced? And this is really where omics is coming in to be such an exciting area in winemaking. And omics is looking at expression of different things. Okay, so genomics is looking at the expression on the gene level, transcriptomics looking at the RNA level, proteomics is looking at what proteins, what enzymes are being produced, and metabolomics is looking at what metabolites are being produced. And essentially what we're doing with omics is we're following the subway map and seeing what is causing certain things to light up or turn dark with response to a substrate. So omics is super duper exciting, and we're starting to see and talk about it much more in everyday conversations rather than just normal super scientific geeky meetings. So what we can see with our omics is we see a lot of heat maps, or this is what we call them, heat maps. And a heat map is a way of taking a ton of data, some very, very large data, and normalizing the data so that we can see what is happening in a very visual manner. So a heat map will look kind of like this schematic. And each lane is representing something different. So for example, my horizontal lanes here may be different yeast strains. And my vertical lanes may be different inputs. It could be glucose, galactose, maltose, maltotriose, phenylalanine, so on, so forth. And we see on the gene level what is happening. On the protein level what's happening, or on the metabolite what's happening, depending what omics that we are actually looking at. And as I said, it's a way to look at a lot of data. And this is looking at data for Sauvignon Blanc and looking at the different aroma compounds that can be produced. And how you read your heat maps is at 0, that is the average. That is where, or the mean, okay? And the mean we can see is kind of this turquoisey color here. Now, if something is being expressed, the first level to the second level are 1 standard deviations, 2 standard deviations, or -1, -2. So essentially we're impacting with a higher z-score, and it's a negative impact when we have a negative z-score. We can see in these different inputs our different response to producing things like ethyl decanoate, hexanyl acetate, damascenone, linalool. Okay, so we're definitely seeing a difference between this pattern, this pattern, this pattern, this, and the control. So these omics are what allows us to really understand what is going on. And we're going to kind of, so some of the information I'm going to show you is based on this type of work. So how do our yeast impact our varietal characterization? How do we optimize the yeast to optimize the varietal characters or our ester? So if we think about these compounds that are produced, and we know they're produced during secondary metabolism because we see the heat maps. So we could really split these aroma compounds up into what we say are aroma impact compounds. So your polyfunctional thiols and your terpenes, and these are generally present at low concentrations. And the individual compounds have a major aromatic impact. Okay, so I might have a low level of thiol or a low level of terpene, but we really see the impact of that organism, that aroma compound. Or we can have our esters, and our esters have a very high sensory threshold. And really, they do not individually contribute to aroma. It is the combination of these compounds that contribute to the aroma. So whereas our varietal compounds have a singular impact, our esters, for want of a better term, have an additive impact. So when we talk about esters, we're stopping talking about individual compounds and talking about groups of compounds that contribute to, for example, citrus or white fruit, yellow fruit, floral, okay? Because it's not just the individual compound. For example, a strawberry. We all know what a strawberry tastes like, what it smells like. But the ester contribution that gives us what we perceive as strawberry is about 20 different compounds, okay? So we have aroma impact compounds and we have additive compounds, and it's the interaction between them all that gives us the excitement from an aroma profile. So last week we mentioned these very briefly. So our thiols, our thiols are organosulfur compounds, so carbon and sulfur, that are found in different varietals. So Sauvignon Blanc is the poster child for thiols, but also found in Chenin Blanc, Semillon, Chardonnay, Colombard, etc. And also in reds like Syrah, Grenache, Merlot, Pinot Noir, our Cabernets, both Sauvignon and Franc. And our thiols are in the non-odoriferous precursor form in the grape. So we don't really smell thiols or experience thiols when we're grape sampling and grape tasting. Our 3MH is found, the compound 3MH, or more correctly, because it's a sulfur compound, 3SH, is found in the pulp. Our 4MMP or 4MSP is located in the skin, as well as our terpenes are in the skin and are not isoprenoids. So in order to extract these compounds, we have to have some skin contact. And this is where mechanical harvesting actually comes in, because it does beat up the fruit a little bit more. So you're breaking down the skin, really revealing these. But if you're picking really cold and you're hand harvesting, then this is where our enological enzymes are key. 6-hour skin contact with our Cuvée Blanc will reveal some of these precursors, or release some of these precursors, excuse me. So these non-odoriferous precursors, 3-MH, this 4-MMP, what are they? How would we recognize them? Well, we recognize them as being tropical or fresh in nature. So our non-odoriferous 3-MH precursor is bound in the pulp with cysteine or glutathione. And when it's revealed from its non-odoriferous to its odoractive form, we pick it up as tropical fruits or citrus notes. Okay. Our 4-MMP, however, still a thiol, found in a different area of the grape in the skin, but bound up with amino acids, so it's still conjugated to amino acids. This represents it as gooseberry or boxwood in whites and as blackcurrant in reds. So again, our thiols, and as we should know, who is responsible for the revelation of our thiols? Who is it that takes from the non-odoriferous to the odor active form? So that is our friend yeast. Okay, our non-sac does a great job with it, our Flavia, but really our Saccharomyces cerevisiae. But because we've been studying thiols and it is a very hot research topic, we now know that for the yeast to take up the thiols in their conjugated form or their bound form to cysteine or glutathione, it does so during the yeast growth phase. So we have to have the thiol there, we have to have it present, we have to have our yeast take it up. And most yeast will take up the thiol. But not all yeast have the ability to release that thiol. Okay? So really it's based on the yeast genetics. So if you're bound up to cysteine, and these seem to be the most prevalent thiols, they're taken up into cell through GAP1 and revealed from their conjugated form due to different enzymatic activity. So remember, the genes control the enzymes. Your glutathione, remember, your tripeptide with cysteine in it, has a slightly different, or has a different uptake system, but it has different metabolic pathways within the yeast in order to cleave off the tripeptide part to reveal the thiol. So it could be cytosolic or vacuolar. So your yeast, not all yeast have the ability to do this. So the key words that we look for when we're talking about yeast that can reveal thiols are thiol revealer, carbon beta lyase, this is the general name for the enzymes that cleave, or previously we called them beta glucosidases or glycosidases. And if we look at the difference in yeast, a yeast like QA23, which has a nice glucosidase activity, versus Sauvie, which has very strong carbon beta lyase activity. And we look at the impact in Sauvignon Blanc from Napa Valley. We see that our Q23 in our orange bar, 79 nanograms per liter of 3-MH, so that thing that we recognize as tropical or citrus, versus our Sauvie, 249 nanograms, versus our 4-MMP, 6.3 versus 97. And then, because I said these are really odor active at low concentrations, What is the sensory threshold? Well, for 3MH, it's 60 nanograms per liter. 4MMP, it is 3. So QA23 does a nice job in tropical thiols as well as these more green fresh thiols with regards to sensory threshold, but Savi does a lot better. And the 3MHA is the acetate form or the esterified form of your 3MH. So our yeast has an impact, and depending on its enzymatic ability, it will give us different aroma compounds. Now, it's not good enough making sure that we have the release of the precursors. We choose our yeast and make sure our yeast is growing to reveal these compounds. Our nutrients has a huge impact on the optimization. And looking at this factorial design, we can see that. So if we're normalized 100, looking at the 3MH concentration, and we can see our vitamins have a positive increase in our thiols. It looks to be about 30%. Our sterols, so our nice yeast membrane sterols, have a positive effect, about 15%. Our oxygen, well, our thiols are very sensitive to oxidation, so we have to make sure we're protecting them. So that makes sense, it is negative. But nitrogen in the form of ammonia is very detrimental to the final thiol concentration, therefore the thiol optimization. So ammonia or DAP, we have a very strong correlation with not getting as many thiols as we would like, and the reason is when we don't have ammonia in excess, while our 3MH is taken up through our GAP1 transport protein. Our GAP1 is our general amino permease transport system. Cysteine is an amino acid, so it's taken up through the system with no problems. The yeast takes the cysteine. We just looked at how it does that through its different enzyme systems. Yeast gets to use the cysteine for what it needs and releases the thiol. Now, if you have too much ammonia present, then we have something called nitrogen catabolite repression, which is a very fancy way of saying the yeast is kind of picky and it's going to pick and choose what it wants based on what is easiest for it to use. So ammonia is easy for it to use. The yeast just gets rid of the hydrogens, gets the nitrogen, goes, thank you very much, don't have to deal with anything else. So in doing so, when ammonia is in excess, it doesn't take up the amino acids through the system, which means your thiols, which are bound to amino acids, are not taken up. So too much ammonia and in the DAP will give you much less thiols. It's something we've known for a long time. So what we do is we are going to look at our nutrition and how we optimize that. So again, we have to have our extraction, our strain selection. You got to handle your strain properly and nourish it. But for thiol optimization, again, I showed you last week how to calculate how much nitrogen is required based on your yeast strain and your sugar. So you calculate what is required, you subtract what you have, and you look at what your supplementation is. If you have to supplement only 50 parts, or irrespective of what your supplementation is, you're using your Go Farm Protect Evolution. And that's so you get the sterols in there for enhanced precursor uptake. At 2 to 3 bricks a drop, we are adding our Stimulus Sauvignon Blanc. And Stimulus Sauvignon Blanc is a newer autolyzed yeast that contains all the vitamins and minerals, as well as some amino acid that helps the yeast take up those precursors and reveals them. And then a third of the way through, we need to regenerate a sugar transporter. So we have different prescription for optimizing thiols, and we generally see that irrespective of the yeast strain that we do, and this was a global trial, so we have Sauvignon Blancs produced in many different countries with many different yeast strains and multiple sets of data that went into this, that we see irrespective of our yeast, we have an increase in 3-MH production when we use Stimulus Sauvignon Blanc. Likewise, when we use our Stimulus Sauvignon Blanc, we see an increase in our 4-MMP. So using the correct nutrient is really going to optimize that thiol. So terpenes, we went over this last week. Your terpene, it could be in the free form. A very low percentage of terpenes are in the free form. And remember, terpenes are citrusy. They could be spicy, floral, and terpenes are bound up to sugars, whereas the thiols, as we just saw, are bound up to nitrogen compounds or amino acids and tripeptides. So if your Saccharomyces yeast has a beta-glucosidase and your monoterpene, which is non-odoriferous, is bound up just to glucose, a yeast with a beta-glucosidase activity can cleave off your terpene and reveal it, so you are able to pick it up. So the key words that we look for when we're looking to express terpenes is beta-glucosidase activity. Now, if your terpene is bound up to more than one sugar, which could often be if it is bound up to 2 sugar, so it is bound up to a pentose sugar as well as a hexose, so a 5-carbon sugar and a 6-carbon sugar, your Saccharomyces has a real hard time cleaving off this bond that links the 5 and the 6-carbon sugar together. But that's where our Flavia comes into play, because it can break that linkage very, very easily. Remember, our Saccharomyces cerevisiae utilize hexoses, but our little Inoculation friends and our non-sac can use pentose sugars. Okay, so that's the reason they're breaking those bonds. So just the different yeast have different genetic makeup, and if they've got the beta-glucosidase genes, they can produce the beta-glucosidase enzyme during fermentation, cleaving your thiols. So esters. Esters, again, these fruity floral notes. Now, depending on the combination of esters you have, you can pick up strawberry or dark fruits. It might be citrusy notes. It could be white fruits like apples and pears, or yellow fruits like peaches, nectarines, apricots. It's a combination of these esters that make them so interesting to us. And these are derived during fermentation. So it is our yeast that is strictly responsible for ester production. And depending on what the inputs to the yeast are, depends on the type of esters that we get out. So the yeast will generally produce ethyl esters and acetate esters. So your ethyl or ethyl esters are produced from carbon being broken down via glycolysis with pyruvate at the end. And to help this part of the pathway, the yeast needs thymine. The pyruvate will be to acetyl-CoA. Acetyl-CoA is a central molecule in yeast metabolism. And for the production of acetyl-CoA from pyruvate, our yeast need calcium pantothenate. If they don't have calcium pantothenate, that cannot happen. And pyruvate is toxic to the cells, so they really want to produce this. And depending on the genes, therefore the enzymes produced that we'll see through our heat maps, we will get a fatty acid CoA. Fatty acid CoAs are medium chain fatty acids, will bind up with ethanol, and that's what give us our ethyl esters, our general fruity notes. Likewise, for our acetate esters, which are generally present in much higher populations, we have nitrogen. Now, not all amino acids, not all nitrogen-based compounds will go through the Ehrlich pathway. It's really the branched chain amino acids, or our aromatic amino acids. And they will go through a transamination, decarboxylation, reduction processes to get higher alcohols. These higher alcohols will again bind with the acetyl-CoA to produce our acetate ester. So depending on how you treat your yeast and what the yeast has as substrates will really dictate your ester production. So ester production, as I mentioned, is impacted by the concentration, excuse me, of the substrates. This acetyl-CoA, it is impacted through adenosine triphosphate, which is a general energy source of the cell. It's impacted by the alcohol levels, both C2 and higher. It's medium chain fatty acids, and all these things are impacted by the conditions. So we want to make sure that the conditions are perfect for our yeast. And some of the conditions that will impact what the yeast is producing is temperature, it is solid content. Lipids have a huge impact on ester production. Some of the latest work that has been done in conjunction with INRA is showing that those natural phytosterols from the grapes, the stigmasterol, the camphosterol, are directly impacting our final ester production. So what they've shown is when they look at 2 milligrams per liter up to 8 milligrams per liter of phytosterols, that the higher the phytosterol level, the lower the ester. Okay, so 2 milligrams per liter of phytosterols doesn't really mean much to us. We're not going to measure these things. Using turbidity is an okay guide. It's not great. So it's like using turbidity to determine filtration success. Sometimes works. But roughly 2 milligrams per liter of phytosterols is about 60 NTU, okay, up to 240. And that's why we have an NTU guide with our recommendations. So looking at
I just wanted to say really quick, guys, it's 11:51. So we've got a number more slides. We'll start answering the questions in about 5-6 minutes if you can hang in there. But it's 9 minutes to noon. Hopefully you can hang in as much as possible because we do have a number of questions we're going to try and hit on. But we are please hang in there. Hopefully we'll get into your question before time runs out. But please, it's a little bit longer than normal, but that's okay because we have a lot of information. So hopefully you'll hang in there. So please do.
Okay, cheers, Darren. The last ones are pretty quick. We've got over like the heavy sciencey ones. So the impact of nitrogen on this aroma index, as I said, we're going to look at the esters together. And we'll see that, again, this nitrogen catabolite repression with when we use too much DEP. And this was a study we did with Oregon State University on their Chardonnay, 2018 now, I believe, 22.4 Brix, 18C. So we see that using our stimulus Chardonnay gives us much more citrus, more tropical, not a huge difference with regards to yellow fruits, but a nice difference in white fruits. So again, I already mentioned the lipids, so I'm not going to go over this. So how do we optimize our esters using our stimulus Chardonnay? So again, we work out how much we need to supplement. We use our Go Farm Protect Evolution, nice, happy, healthy yeast, nice, happy plasma membrane. We adjust our nitrogen if needed at 2 to 3 Brix drop. And we add our stimulus Chardonnay 1/3 of the way through, and that is our aromatic amino acids. So our phenylalanine, our isoleucine, our valines, for ester synthesis, as well as those essential vitamins, the biotin and the calcium pantothenate, which helps with the pathways. So we could really use our yeast, the information we have about our yeast, to shift things in a thiolic way if we're working with a thiolic cultivar, or esters if that's the style we want to do. And this is exactly what we did in this Gruner Veltliner trial from the Finger Lakes. Exact same fruit split into 3 lots. One lot got Stimulus Chardonnay, that's all the orange bars. Another lot got Stimulus Sauvignon Blanc, and the third lot was Fermentation, actually. And we looked to determine what the aroma impact was, or what aromas were produced using the different nutrients under as similar conditions as you possibly can have. And we've seen that the sum of esters and acetates increased when we used Stimulus Chardonnay compared to the Sauv Blanc and the Fermentation, as we would expect. We saw that when we used the Stimulus Sauvignon Blanc, we had a huge increase in our 3-MH, as we would expect. And then our 3-MHA, so the esterified or the acetate form of our 3-MH. And we've seen a little bit better with our Stimulus Sauvignon Blanc. So everything is above sensory threshold, even with our Fermentation. But taking that one fruit, splitting it into 3 different ways, we can have 3 very different wines. And I hope that helps to show you how we can use nutrients to really drive wine style. So we've spoken a lot about varietals and enzymes in, or esters, sorry, and all the enzymes involved in white wines, and it all goes for rosés as well. What about reds? Well, this year we're actually going to be doing another year of trials in the North American market on the stimulus for reds. Stimulus Syrah, which increases 4-MMP production. So it's helping the yeast produce that carbon beta lyase, so that we're really enhancing that blackcurrant bud aromatic. And Stimula Cabernet, which is increasing the esters, and we're seeing it in the black and the red fruit esters. So it's not just that we've been focusing on the whites, just it takes a much longer period of time to really study the aroma impact on our red. So that's really exciting. So we spoke about yeast and nutrients, a little bit about lipids, a little bit about temperature, a little bit about skin contact, but all of this rolls into driving wine style. So we want to get the precursors out, or want to produce aromas, and we want to stabilize them. And a lot of that information is in our wine style guides that you could find on our website. We have wine style guides for aromatic whites like Pinot Gris, Verdeo, Chardonnay, our Rhone whites, rosés, depending on what style you want your rosé. Do you want it varietal, tropical, ester-driven, red fruit style, and of course, Sauvignon Blanc. So with that, lastly, our non-sac will drive aromas. Our Flavia will drive both terpenes, norisoprenoids, and thiols, okay, because of its advanced enzymatic activity, its ability to cleave the pentose, and its ability to produce the carbon beta lyase. Our Biodiva, the Troilspora delbrueckii, produce a lot of esters and a lot of polyols, specifically a rabanol, which is a C6, which gives you that mouth feel, and Lactea producing your lactic acid for freshness. And then we can't forget our little friends, Inococcus, who are the organism of choice for conducting malolactic fermentation. There are places in the world that will use Lactobacillus plantarum to produce or to conduct ML, because it doesn't produce any VA from sugar. It is homofermentative. But our MLB are involved in aroma production as well. We really talk about them as being diacetyl producers. But as you can see from these aroma charts, these pictograms, we see that they're producing things like esters. They are enhancing varietal compounds as well. So, you know, there's a lot to driving wine style, and the more conscious your decisions, the more impactful you can be. So we've gone over inoculation rates. We have gone over the importance of proper yeast rehydration and how to optimize aroma production by yeast using a lot of scientific data. We are a very science-driven company, so for that, I don't know if I should apologize or not, but it is the science that we like to bring to you through these webinars. So with that, thank you so much. Thanks for hanging in there. Thank you for taking time. Have a great harvest, and let us know how we can help. And over to Darren.
All right. Yeah, no, don't apologize. I mean, we need that information. I mean, there's so much, like, if you go online and you go on like these blogs and these websites, not these, I sound like I'm 75 years old, but when you go online, you try and find answers to these questions. It's like, you know, garbage a lot of times. I mean, the stuff that I read, it's like, wow, like they don't even, there's a lot of confident people with an internet connection. And that sometimes drives folks in the absolute wrong direction. And I can tell because, you know, they'll ask these questions, you know, and you can kind of see just by the nature of the question, how, where, just weird questions, like knowing something, do you say this? And I'm like, well, that first part you just said doesn't make any sense. So it's really, really important that we keep folks. If it seems like it's a lot, you can always go back and listen to it again. I mean, we're not trying to replace, you know, university degrees. We're not trying to place that, but we do want to make a point with these first two webinars to get us at least all on the same kind of level of information. So then we can start to debate about whether, you know, some things help things and versus others. So it really is very thankful that you spend some time in the webinar. Again, we are 12:01. We are at the end of the material. If you need to go, I understand that. Thank you for showing up. But we're going to go ahead and run through these questions. It's the lightning round because I don't want to hold folks too long. When we have one hand up, and I want to allow that person to talk first. Michelle, if you would still have your question, I'd love to hear from you.
And you are on mute.
Ask to unmute. I'm asking. It might be a mistake too. Sometimes people raise their hand
and just didn't put it down. Okay.
Okay. Well, that's fine. We'll move on. So the first question is from the post earlier today. Is right when you were talking about rehydration, and it says, "I understand lowering 15 degrees Fahrenheit in 15 minutes. If I had to drop 30 degrees Fahrenheit, you know, their must is at 30 degrees lower than what they were at. Do I need to take 30 minutes?" And what is the maximum rate that I can lower the temperature before adding to the grapes? I mean, how many degrees do you have to worry about before you're shocking? How much temperature can you change without shocking the fruit?
18 Fahrenheit.
I mean, shocking the yeast. 18 Fahrenheit. So
18.
Shocking the yeast. Okay, so make it clear.
So that's why we say 15 for 15 and repeat it as many times as you need to. Take the time up front. It's way better than having a sluggish fermentation at the end.
Okay, so just make it clear for everyone. If you're rehydrating yeast and you're lowering it from that 104 degrees down to the 50 degree Fahrenheit juice that you have, you cannot change the temperature more than 18 degrees at a time. 15 for 15. That sounds like a very simple way of putting it.
Fahrenheit.
Fahrenheit. Yes, not Celsius, everyone. Fahrenheit. Hopefully your juice is not at 50 degrees Celsius because that's a little hot unless you're trying to do some kind of flash. You mentioned the quality of water. Are there any recommendations on the max hardness?
Normally we say 200 ppm.
200 ppm.
That's from memory, but that's what I
No,
think.
no, that's cool. No, that's great. If I
200.
press grapes directly, I believe I will get 3 mH from the pulp, but no 4 mmp. Is that correct?
It depends if you've got 4 mmp. So these varietal characters sort of depend on your clone, your viticultural practices, your nitrogen and sulfur supplementation or spray regime in the vineyard. But if you're not having any skin contact, if you're going straight to press and pressing quick, you will not release your 4 mmp.
Oh, okay. So yeah, great. Okay. So if you're going for that style, don't do crushing, don't do destemming, go straight to press. But if you do want some of that 4 mmp, you know, at least you might even consider using enzymes for that. I mean,
20 grams per ton of Cuvée Blanc, 6 hours. That's what I do.
Yeah, you know, and that's one of the things I noticed years ago when I first started, when we had, 20-ton presses, and I'd go out and I'd smell the pumice of the white fruit, and I could just smell all that fruit, all that flavor just kind of disappearing, moving away. I really, really wondered what you could do with the wine. And I did have a friend eventually that fermented his Sauvignon Blancs on the skins for a week, and then he used a bunch of PBPP and casein to remove the bitterness. And I tell you what, it was a very interesting wine. Very interesting wine. How do you correlate excess ammonia with optimal yan? Is there too much ammonia sometimes? How do you correlate that?
In general, your yan is, no, this does not hold true for Washington, but in areas that have had agriculture, you're generally about 2/3 to 1/3 ammonia to amino acids. So generally, I don't want to see more than 100 parts ammonia. The lower the ammonia, the better. In fact, it's much easier to manage low yan ferments than high.
What level of nitrogen is in excess? Like what's your limit where you're hoping you're not any higher? Do you have a hard line?
I don't want to see more than 250. 150 is what you need. If you're 23 breaks, you're using a medium to high yeast, nitrogen requirement yeast 250 is sufficient.
Okay, cool. Excellent. Do you consider Fermate O better than Fermate K, or could you use a blend of two? Could you use one and then use a blend of two?
Fermate K is fantastic. It's what we call a complete nutrient. It's been around since 1985. It came out before we had our Go Farm Protect Evolution, or Fermate K. So we want to make sure that these have the vitamins and minerals. So I would much rather replace my Fermate K with Fermate O, making sure I'm using my Go Farm Protect Evolution during rehydration, unless my yan is too low. You'll notice in the supplementation guide that we have in the handbook and online, we still do include Fermate K when we have at the second nitrogen addition, so a third of the way through, when we have to supplement more than 50 parts and more than 100 parts. So there's still a role to play for Fermate K, but we are much more focused on the organic nitrogen because the yeast gets so much more out of the amino acids.
What would be the impact of using Stimulus Chardonnay in Sauvignon Blanc?
You'll get more esters.
Oh, that's it. Okay. How do the Stimula Cab and Syrah differ from Booster Rouge?
Well, they're all different yeast that we're using to make these. So their base is different. How they're produced, so what they're giving back out when we add them is very, very different. So the Stimulas, we're really focusing on the vitamins, minerals, and very specific amino acids. The boosters, likewise with the Optis, we're focusing on polysaccharide release.
Okay. Any experience with your various nutrients and ferments of melomels and piments, which I think melomel is kind of like a mead?
Not got a clue.
Oh, it's a blend of cider and mead. Well, cider and mead sometimes, right? Nutrient deficient?
Yeah, especially the mead portion. So it's essentially like we're fermenting a seltzer, but if you at least put the cider in it, you're getting the phytosterols in there. So I would still follow good fermentation practices by looking at my sugar, my yan, supplementing accordingly.
Okay. Can flavia be used during settling of juice? Is it vigorously fermentative?
It is not vigorously fermentative. If you add it during settling, I would worry about it settling out and not actually being able to do what we want it to do. So I would much rather add it just post settling and let it work for 24 hours before I go in with my Saccharomyces.
Okay, sounds good. If my must varies in sugar from 22 to 27 Brix, can I stock Go Firm Protect only, or do I need to have both Go Firm and Go Firm Protect Evolution?
Nope, just go straight for with Go Firm Protect Evolution if you're at 15 Brix or 27 Brix. It's actually got much more sterols in there, better complement of vitamins and minerals, so it's going to help the yeast tolerate the high alcohol or the low pH. So it's just a much more advanced product. Go Firm Protect Evolution. That's why we work with Lallemand, because they continue to evolve their products based on knowledge, research-based knowledge, and winemaking needs.
Okay. So here's a summary, kind of. Would you agree with this statement? Wait, what?
You're laughing. I'm like, oh gosh.
No, it's all good. So with proper nutrients, generally higher firm temperature leads to more thiols, and lower fermentation temperatures leads to more esters.
I don't know. That's ambiguous. What is higher temperature? So you're trying to get the temperature optimized for enzymatic production. Generally, you'll notice in the thiol work, the temperatures are wavering between 65 and 70, so 18 and 22. And that's for our carbon beta lyase production. For esters, you'll notice in the work that's done with Dr. Murray that he starts his low temperature for esters at 20 Celsius. So if we're down in the 50s, generally we have more isoamyl acetate, more ethyl acetate. Once we start to get into the 60s, we have more of the fruity esters. And once we're into the 70s, we have more floral. Now they are highly volatile, so then it's the stabilization, which is a different conversation from production.
Yeah. I mean, there's so many things are determined on these. I mean, when you talk about all the different parameters, just trying to stabilize several of them. It's interesting for years now, when people ask me about a particular yeast and its performance, I'm still go back to those basics. Like, well, can you control your temperature? Do you know what your YAN is going to be? Do you have any idea about your ethanol tolerance or your potential alcohol? And if they can't answer, one, if not three of those questions, I'm kind of like, well, I can't really make any promises about the yeast performance because you don't even know I'm not entirely sure you can control these major parameters. One last question. Somebody noticed that Lallemand has begun printing glutathione contents on their packages. Is that something that we're going to post on Scott Lab's website and put in the calendar? Is that something that people, if they're interested, just go to Lallemand for?
Go with Lallemand. That is part of the new OIV monography, that if something is glutathione, it has to have a certain number or a certain ppm of reduced glutathione, which is the active form, and not the bound up sustainated or oxidized form. So that is, they're publishing them because they're a very transparent company. That has to do with the OIV regulations.
Okay, so that was part of some of the grumblings I heard before where the word glutathione, it's kind of like the word energy. People were like, I got glutathione, but there was no real, it's very like the OIV to be like, no, you can't use this word, energy and say that you've got lots of it. Is that basically what they were trying to deal with?
Yeah, they want to make sure that the amount of glutathione that is there is an actual active glutathione, and the active glutathione is the reduced glutathione. So you've got oxidized compounds and reduced compounds, right? So yeah, that's all it is. It's part of the European regulations.
Yeah, I want to make that point too. When we talk about Lallemand and when Scott Labs talks about, quality of products, it's not just something we just pull out are vague. You know, it's very clear that they have to follow OIV limits and regulations, and you know that just requires a whole nother level. So you can't just buy like a bag of stuff and then say, hey, it's my nutrient now. You can't do that and hope to trade with any nation that's part of the OIV. And we choose as a supplier to follow those guidelines, but not every supplier does that. I mean, any Yahoo could go out there and buy a bag of, you know, yeast hulls and tell you it's a nutrient, correct? In the United States, right?
Yep.
Yeah.
Well, that's the challenge is working with a supplier who you trust, and also working with Lalaman because they are a producer of these products, and they produce a lot of products for human consumption. So their regulations are so stringent and so tight on the production level that they're actually exceed OIV regulations because of those stringent regs that they go by.
That's a relief. At least I know I can go there. You know what I mean? At least I you know, there's a number of suppliers, and it's like there's a lot of different suppliers, a lot of different products now, and it's nice to know that you have at least one home, or we're pretty much, you can at least rely on that as something you don't have to worry about. So some of the results for the polls, here's the first poll. We got 40 votes total, but the vast majority, after fully rehydrating your dried yeast, how do you add your yeast slurry to your must? 60% said I mix fully into the must juice. 20% said I pour it into the corner of the tank and leave it. And 20% said I incorporated the yeast at the next pump over or punch down. And then no one said I don't add any yeast. It's probably not surprising considering that this is a presentation about adding yeast. Poll number 2, what type of nutrient do you use? Here we go. Rehydration, Go Firm, Protect Evolution, etc. 75% of the folks that answered the question said that. 53% they use a complex nutrient. 58% used an organic nutrient. 38% said DAP. 23% other organic. And then 3% who are watching this webinar today said, I don't use nutrients. So that's always interesting. And then wine style drivers. Of the following, what do you think what drives wine style the most? 45% said yeast, 15% said nutrients, 5% said temperature, 13% my attitude, but coming at number 2, when asked if the most following, what do you think drives wine style? 23%. The number 2 answer was your accent. And I've got to say, I mean,
what can I say? We're clowns. As you can tell, Darren and I take this seriously.
It's good to know. We want, you know, we're always continuously improving. You know, I sound like my nose is always pinched. And you can't do much about your accent. I love your accent, though. It's fantastic. And I'm glad people are making decisions based on your accent. That's very nice to hear. All right, guys. So that's it. So what can I say? Thank you so much for your time. I can't stress that enough. I know we went over a little bit today, but we actually came in pretty nice. Don't hesitate to reach out if you have any other questions or comments. We believe in continual improvement. Thank you again for your time. I can't stress that enough. My email is darrenm@scottlab.com, darrenm@scottlab.com. We've posted the webinars on our website, so just Google Scott Labs webinars. It should show up in the search results. Thank you all so much for showing up. All of us at Scott Labs want to wish you and your families health and happiness. Any final comments, Nichola?
Nope. Have a great harvest. It's creeping up here pretty quickly. Stay safe.
Freaky. Harvest every year. We get the harvest shakes, and they're starting to kick in. Thanks, guys. Hang in there.